First, we've got a presentation from Dr. Katy Thomson, who works within the IOI. And she's going to be telling us about, so she's the lead of the BARNARDS II study, which looks at antimicrobial resistance and neonatal sepsis. And I think she's going to give us some early insights into the preliminary results that they've been getting. So thank you very much.
Can you hear me at the back? Yeah? Okay, great. Okay. Good morning, everyone. Thank you for having me today. So I'm Dr. Katy Thomson, and I work within the Ineos Oxford Institute of Antimicrobial Resistance. And I'll be giving some very preliminary findings from the BARNARDS II study, looking at antibiotic appropriateness and outcomes in neonatal sepsis. And just a brief trigger warning, there are discussions of neonatal mortality in this presentation. So what is sepsis? Sepsis is a life-threatening body's reaction to a bloodstream infection, and this can be caused by fungi, parasites, viruses, but most commonly bacteria. And by neonatal, it means within 0 to 28 days of life. So it's estimated that between around 1.3 to 3.9 million cases of neonatal sepsis happen globally every single year, and accounts for up to 25% of all neonatal deaths, with a very high burden. As you can see in the graph on the right, it shows a very high burden within low and middle income countries, particularly Africa, shown by the darker colours here on the map. So the clinical pathway of sepsis is that the neonates will display a range of signs and symptoms. These symptoms can be extremely vague, so they can be fast heart rate, slow heart rate, fast breathing, low breathing, so it's very difficult to diagnose and can be due to other ailments. So after the clinician suspects sepsis, a blood sample is taken, and then the neonates are prescribed with empirical broad-spectrum antibiotics. The World Health Organization recommends ampicillin and gentamicin for this for neonatal sepsis as a first-line treatment. And then the blood sample, the ones that they found a pathogen in, so the positive blood samples, then they'll run diagnostics in the lab. So looking at species ID and susceptibility profiling. And then from these results, or the clinical response of the patient, then clinicians will choose to escalate or potentially de-escalate antibiotic treatment. So some BARNARDS core aims that are relevant to this presentation are to quantify the burden of neonatal sepsis across 13 hospitals. We've got 5 in Nigeria, 5 in Pakistan and 3 in Bangladesh, and the pictures are the country PIs plus the UK country leads we've got within the Oxford Institute. And then we're also looking at risk factors for development of sepsis, development of a resistant infection, and also of mortality. And then we want to also look at fully evaluating antibiotic use. There's very sparse data in this area, particularly in low income countries. So the methods for the BARNARDS II project, we've got the hospital sites, and then the neonates and the carers are enrolled, and a wealth of clinical and admission data is collected by research assistants at the site. And then neonates with diagnosed sepsis. It's not working. Sorry. Is this working? I was on mute. Okay. Sorry. Oh, wow, okay. Okay. Thanks, sorry about that. I hope you heard me. Sorry if I didn't. I'll carry on, but if you've got questions about the first bit, let me know. So after the neonate's diagnosed with sepsis, then the site laboratories will do, as I've mentioned before, the species ID and the AST profiling, antibiotic susceptibility testing, and then throughout the entire admission, the research assistants are collecting all of the antibiotics that are administered throughout the whole admission process. We then collect in-hospital outcomes, so whether the neonate was successfully discharged or, unfortunately, if they passed away before discharge. And then those discharged will also have a 28-day follow-up by a phone call just to check how they're doing. So the neonates with positive blood samples, then those isolates that are grown from the blood are stored and then shipped to the UK, where we do further characterization, including species ID through MALDI-TOF, minimum inhibitory concentration testing, so looking at the specific breakpoints of the bacteria, and whole genome sequencing as well. And in this talk, I'm talking about appropriateness of antibiotic therapy, and this we've defined this in this presentation as a pathogen that's susceptible to at least one antibiotic that is administered to that neonate. So some preliminary numbers on this slide here. So we've got a total so far of 14,624 neonates with clinically diagnosed sepsis. The BARNARDS II project, the enrollment has finished now, but we're still cleaning data, so these aren't necessarily the final numbers. They're going to change slightly, I think. And out of these neonates, we've got antibiotic data for 14,259, so most of them were given antibiotics, and just over 5,000 actually had culture-confirmed sepsis. And then we've got from all the 5,000 with culture-confirmed sepsis, we have appropriateness data, so MIC data plus antibiotic usage data for just 2,300 bacterial species, as well as 508 fungal isolates. Due to antifungals aren't given commonly as a first-line therapy, so this presentation is mainly focusing on the bacterial, but I'll mention fungal again a bit later. So overall, with the bacterial species, we've got just under 2,000 with appropriateness data and outcome data. So just to give you a bit of an idea about the kind of antibiotics that are given at the sites, we found a range, I think it was around 200 different combinations of therapies. We found that most neonates were given 2 antibiotics as a first line. So I mentioned earlier the WHO treatment is ampicillin gentamicin. The most common therapy given at the sites was cefotaxime and amikacin. And then 3 antibiotics were given to some neonates. Just over 10% of neonates received 3 as a first-line treatment, and some neonates also only received 1 antibiotic. And we saw antibiotic escalation in 3,500 of the neonates, so not all of them received anything beyond the empirical first line, and some of them were given up to 6 lines of treatment. And surprisingly, we found that only 40 neonates out of over 14,000 were actually given the WHO recommended first-line treatment of ampicillin gentamicin, which may seem very shocking. But looking at these, we know that these hospitals have an extremely high burden of antimicrobial resistance. And so in the next slide, we can see why this might have happened. So these are minimum inhibitory concentration profiling done through agar dilution by research assistants, pictures in the bottom right there. And we found extremely high resistance throughout nearly all of the antibiotics that are tested. And we can see the 2 antibiotics highlighted in the box. It's ampicillin, so this is for the gram-negative bacteria, and gentamicin, just to the right there. And we found nearly 100% of resistance against ampicillin and around 70% of resistance to gentamicin. So this is why it's not really effective in these settings. The WHO also recommends third-generation cephalosporins as a second-line treatment. And again, we see extremely high resistance, up to 90%. When you have an inhibitor with the cephalosporin, it's a bit lower, but we can see that it's still around 70%. So this is why ampicillin gentamicin is not really being used on the ground. And we see lesser extent of resistance to gram-positive. So the resistance morphologies is in red. And again, we found less of an issue in the gram-positive bacteria, but still ampicillin resistance is 80%, and gentamicin is 60%-ish. So when we looked at what pathogens cause a neonatal sepsis, we can see that in the majority of cases are actually due to the gram-negative bacteria, which has shown extremely high resistance, which composes 47% of the cases. And then the gram-positive bacteria cause 39% of cases. And then we've got a smaller section, which is fungal pathogens, which is actually causing 14% overall from the 3 countries, which was mainly predominant in Bangladesh sites. We found that when we look at the appropriateness of treatments that were given, no fungal infections actually received appropriate empirical treatment. It's because antifungals isn't normally prescribed first line for neonatal sepsis and was only administered first line in 47 cases overall. That didn't happen to be fungal infections, unfortunately. So when we look at the bacterial isolates, ampicillin and gentamicin, the first line recommended therapy, would have been appropriate for 24.9% of the isolates. And then the sites rarely use this, but they obviously were having a lot of difficulties selecting correct treatment because they don't know what the pathogen is when they prescribe the empirical therapy. But they managed to achieve 44.9% of appropriate first-line treatment. So this is actually much lower when we look at the gram-negatives. So 28% of the gram-negative cases received appropriate treatment, and this is higher for the gram-positive. So this is just an alluvial diagram, basically just displaying what I've just said. So we can see the red box on the left shows inappropriate first-line treatments, which is slightly bigger than the ones that received appropriate treatment. And then we can see that no fungal isolates received received appropriate treatment, and then most of the inappropriate were actually in gram-negative, and unfortunately a lot of these ended in mortality. But we did see some inappropriate first-line treatments that had a more positive outcome, and I'll talk about that a bit in the next few slides. So when we looked only at were they given appropriate first-line treatment, yes, or appropriate first-line treatment, no. So alive is in blue in the bar chart and deceased is in red. And we found a significantly higher amount of mortality in those that were given an inappropriate first-line treatment. So this was shown to be significant when we did unadjusted analyses. But then when we looked at multivariate analyses, so I mentioned in our methods slide that the research assistants on the sites have actually collected a wealth of clinical data, so looking at the birth factors of the neonate, why they were in hospital, where they were born, what mode of delivery. So we've got a wealth of clinical information that we're currently kind of cleaning and going through at the moment. So when we did the adjusted analyses, we found that whether they were given an appropriate antibiotic, yes or no, was actually not significant when we accounted for the other confounding variables. It was still related with a low mortality, but due to the high confidence intervals, this wasn't found to be significant so far. The most significant associations we found with outcome was gestational age, or higher gestational age is showing a better outcome, and also gram-positive isolates compared to gram-negative isolates. So it could be, so obviously a lot of the neonates that are admitted to the hospital, a lot of the premature neonates will be, a lot of them unfortunately do get nosocomial hospital-acquired infections, and these are more commonly gram-negatives with a higher resistance profiles. So there's a lot to unravel there about what's actually going on at the sites. We also found in these results that C-sections were actually associated with a better outcome than those neonates that were vaginally delivered. But when we look at the data again, I think it's more about the referral pathways, and more C-sections were born at a BARNARDS II site, so they had better access to healthcare, and probably were seen quicker by a doctor compared to those that were born off-sites. So this slide was still unraveling the complexities of the data to really grasp what's going on here. But I wanted to show this slide to show the importance of collecting confounding variables when carrying out research projects, and need to understand the context fully when developing studies and analysing data. So this is really complex, and we're still undergoing further analyses. So I've talked so far about first-line treatment only, so we're starting to look at escalation practices as well. So this is a Sankey diagram just showing the appropriateness. So appropriate first line is shown in blue, and inappropriate is shown in orange in this graph. And then you can see if the gram-negative is in or positive is in the second line, and then we can see whether these neonates had escalated to a different antibiotic treatment regime. And we've seen that most of them did not. Were not escalated, and this might be due to premature. It might be that the neonates unfortunately passed away before they were able to be escalated. So we can see that those that had inappropriate and weren't escalated, most of them unfortunately did. A higher majority did unfortunately pass away, but we can see that more neonates that were given appropriate first-line treatments were not escalated compared to those that were given inappropriate. And sorry, I've got myself confused. And yes, but we can still see that some that weren't escalated and given inappropriate treatment still had a discharge, still had a positive discharge and was successfully discharged from hospital. So we need to look at this data a bit further. So the next steps is we want to split patients into early onset and late onset sepsis. We want to look at exactly the timing of the antibiotics, the time to appropriate antibiotics, looking at the dosing as well. And at the moment, we've looked at resistance versus susceptibility, but we want to look at the specific breakpoints of the bacteria. So it might be that the neonates that survived with inappropriate antibiotics, maybe their pathogen was on the breakpoint value, so maybe actually the antibiotic was effective. And also we want to look at their whole genome sequencing data, so looking at virulence genes and seeing whether they were pathogenic. It could also be that some of these isolates are potential contaminants. So we're still trying to unravel kind of the web of this complex data, really. So yeah, just thank you to the Ineos Oxford Institute for funding this research. And we've had a huge team working on this, so everyone at all the sites, they've worked really hard, and everyone within, we collaborate with Cardiff and in Leiden, and I know that there's a lot of people from the BARNARDS team here today as well. So, and Kirsty, the lead for the surveillance program, and Tim Walsh, the PI, and Joe, and yeah, there's lots of people here today that have contributed to this project. So it's really great to see it coming together. I think that's it from me, and thank you so much.